So I think that the opportunity is unique, no? In the sense that there has never been so much information. Now the question is how to integrate that information and make good use of it, no? And that's the other side of the coin, that there is also a huge amount of effort in integrating data and connecting data. So the harvest is going to be even bigger than the data itself, no? And this is going to come. And what is needed is people that will be able to also deal with big data, that will handle this data and will see that not only that you have to learn how to deal with it, but to have to find ways, new ways of analyzing the data in ways that integrates the different aspects of it, especially in biology, where you are going to, everything is integrated. Even though we break it to study it, the reality is that everything works together as a system, and we have to think like system engineers, no? And that's the way to really move forward.
At the end of the day, we believe that is a good thing, because evolution has co-opted those elements for novel function and has fixed this into the genome in a way that even if it's completely invaded by these elements, you have a lot of specificity on how the genome is using these elements in different cell types. And they could respond to different transcription factors, they could be chromatin RNA, that could be stationed for genome organization. But at the end, the challenge is that to being able to decode all these signals in a specific way and understand how they contribute to the transcriptome, so at the end of the cell identity. And is what now we are called to do, because with the last genome release, we could see this signal very well, but still we need to develop a lot of technologies to being able to dissect specifically this signal bioinformatically, also in molecular biology. And I really believe that we will succeed this in few, or maybe not few, years.
Yes, I do think it's the revolution, and I think it's ongoing, and I think there is even more to come. So really single cell allows us to look at the single cell resolution, meaning that we can, as like you said, look at the heterogeneity of the cells and start looking at the differences between cells and how they behave in a normal versus disease situation. And as the time passes, like our ability increases, like from going from initially looking at the transcriptome of the individual cells to now looking at the epigenome, combination of these two. And now with the development of AI, I think this is going to even move forward. So I think that we are actually now maybe at the beginning of the real thing coming up. I think we should be generating even bigger data, with more modalities, and then start incorporating AI and start kind of seeing the patterns and really kind of to understand the mechanisms behind normal processes. In my case, we are looking at the development, but also in terms of the disease.